Hey everyone, welcome back to Windows 8 Studies. I hope you all are doing well. So we were learning about cell biology in which we discussed about cytoskeleton. So we discussed about actin filaments, which are also known as microfilaments, intermediate filaments as well as microtubules in detail. So today we are going to discuss about plasma membrane. So we will be discussing about plasma membrane in 2 to 3 lectures. Today we'll be only discussing about the different models that gave us an idea about what is the composition of plasma membrane and what is the structure of plasma membrane. So today's discussion is kind of an history lecture where we will be going through the different ideas that were given by different researchers at different point of time to introduce that what kind of structure possibly plasma membrane could have. So let's get started with the lecture. So I've listed down the different models here and we'll be going through them one by one. So the very first model I'll be discussing is Overton model. So in this model, the experiments by researchers, they were done on the plant root hairs. So when researchers were doing their experiments, they observed that the hydrophobic molecules, they can cross the plasma membrane easily. So on the basis of the idea that like dissolves like, means the polar molecules can interact with polar molecules and non-polar molecules they can interact with non-polar molecules. From this idea they concluded that whatever plasma membrane is made up of, that molecule should be hydrophobic. So they gave the conclusion that plasma membrane is made up of lipids, because from all of the biomolecules, lipids are the only structurally integral biomolecules which are hydrophobic. So they concluded that they are made up of lipids. The plasma membrane is made up of lipids. But in this model, they did not mention anything about the structure. So they did not give any idea about what is the composition of plasma membrane, like even if it's made up of lipid, what kind of lipids it is made up of, and how they are arranged in a membrane. That wasn't clarified enough, and that is why this model was rejected. Then came up Irving-Langmuir model. So this model stated that okay, plasma membrane is made up of lipids, but they said that they are arranged in a monolayer. So this is kind of improvised model. So this gave a bit idea about what is the structure. So they said that they are they are arranged in a monolayer. So this is how a lipid molecule looks like. It has a hydrophilic head group and a hydrophobic tail group. Okay, so let's discuss about lipids for a second. Okay, so let's discuss about lipids for a bit. So the very simplest form of lipids is known as triglycerides. Okay, so triglycerides are the simplest lipids. Okay, so they are a special class of lipids. Lipids contains multiple fats and oils, but these are the simplest ones. And let's take a look at what is the structure of triglycerides. So you must know this molecule. Okay, so 3 carbon chain with each carbon having an hydroxyl group. What is this called? So this is a glycerol molecule. Okay. So this is a glycerol molecule. While what if when on the glycerol molecule, when each hydroxyl group, the hydrogen is replaced by a fatty acid, then that forms triglyceride. Okay, let's take a look at the molecular structure of fatty acid. What is how the molecule looks like, the fatty acid. So fatty acid, it's an acid. Okay, so it should have a functional group, acid. Acetic acid has COH group, right? So this acidic group should be there. Carboxylic acid. So fatty acid represents the carboxylic group in the fatty acid. So and it is fatty acid means a long hydrocarbon chain should be there. So we are representing the hydrocarbon chain by R. Then it is it is paired with CH2 and that has a carboxylic group. So this is a structure of fatty acid. So depending upon the number of carbon that R has, the fatty acid will have it name. For example, the 16 molecule, the carbon with okay, so it's having the fatty acid with 16 carbons is known as palmitic acid. Okay, or if it has 18 carbons and one double bond, it is called oleic acid, linoleic acid with 2 double bonds. Okay, so C18 delta 9 and delta 12, it is known as linoleic acid. So depending on what is the depending on the R group, what the how many carbon it has, whether it has double bonds or triple bonds, depending on that the fatty acid differs. Okay, so this is how a fatty acid looks like. So when the hydrogen of the glyceryl, each hydrogen of hydroxyl group, okay, so there are three hydroxyl group in glycerol, so three OH groups. Okay, when the hydrogen from each OH group is replaced with a fatty acid chain, then it forms glyceride, triglyceride. That is why it is called triglyceride. Okay, CH2. So this O, it has oxygen. It has oxygen. So the reaction occurs between the acidic group and the oxygen. So what will happen that it will have the acidic group C double bond OO, and it will have its own fatty acid chain R2 C double bond O R3. Okay, I'm considering this as whole an R dash group. So I'm representing it as R1, R2, and R3 to represent three different fatty acids. So we are assuming that three different fatty acids have been attached to this molecule, to this glycerol. So this is triglyceride. Okay, so this triglyceride molecules is the lipid. So in this lipid, the glycerol backbone, when to the glycerol backbone, let's say that we remove this fatty acid. So instead of this fatty acid, the phosphate group is added. Okay, let's say that instead of this fatty acid, a phosphate group is added. So how will it look like? So let me restructure it a bit. So CH2. So this will have its fatty acid group. It will have its own fatty acid, and this, instead of, we are removing this and we are adding a phosphate group. Okay, so let me just rewrite it here. Oh, minus. So this is how it will look like. So this phosphate group is hydrophilic, well, this is a backbone. This is glycerol backbone, and those long hydrophobic fatty acid chains. So these are long hydrocarbon chains. They are the hydrocarbon chains. They are the hydrophobic ones. Oh, hydrophobic. So they are, because they are very long, so they are often called as tail. So they are the long tail groups, the hydrophobic tail groups, and the phosphate is going to form the head, which is hydrophilic, and that this is how the structure looks like. So this will be the hydrophilic head, and this will be long hydrophobic tail. Okay, so I hope the composition, like the molecular structure of lipid, is clear to you all. So how the lipid looks like? Lipid has an it has hydrophilic head, and it has hydrophobic tail. Okay, so they said the Irving Langmuir model, they said that they are arranged in a monolayers. So this means that this is how the lipid structure will look like. So this is how they are arranged in a monolayer, and that's what plasma membrane looks like. But this model has a fundamental problem in the explaining the stability of the plasma membrane, because inside the cell, like extracellular matrix as well as, okay, as well as cytosol, both of them, they are hydrophilic, right? So this could not explain the stability of plasma membrane, because cells are stable. Plasma membrane is a stable entity. It is not, it is not forming abruptly and it is getting disrupted abruptly. So it is a stable entity, but this, when you say that it is monolayer, it actually questions that stability. So it could not explain the stability of plasma membrane, because this actually this model, it is quite unstable, because hydrophobic tail cannot interact with the hydrophilic cytosolic part. Or even if you know, if even if there could be two possibilities of being in a monolayer, either Either this is cytosolic side, so either cytosolic side has the hydrophobic tails, or the extracellular matrix has the hydrophobic tails, and let's say that this is cytosolic side. In both of the cases, even if it is extracellular matrix or cytosolic part, in both of the cases, hydrophobic tails cannot react with the hydrophilic environment, because cytosol or extracellular matrix, both of them are hydrophilic. So this model was again rejected, because lipids, they are amphipathic molecules, so they need a stable, a stable structure for maintaining the stability of plasma membrane. So this arrangement of being a monolayer was rejected. Then came Gorter and Grendel model, which said that membrane is a lipid bilayer. Okay, now, so this was a very interesting idea of having an amphipathic molecule being, being structured in bilayer. So they said that hydrophobic tails, they faced each other. So this, they said that those hydrophobic tails, so these are the hydrophobic tails, they are facing each other. But again, this model was rejected, but because it could not explain the other properties of membranes. Like, we know that certain membranes are selectively permeable, while certain membrane allows the passage of molecules freely. Okay, so certain of them, few of them are completely permeable. For example, the outer membrane of mitochondria is fully permeable to the molecules, while the inner membrane of the same mitochondria, it is highly selectively permeable. So this different kind of, properties of membrane that were not explained. So even though Gordon and Grendel model solved the problem of the stability of, they explained the stability of membrane. So like, they explained that membrane is made up of lipids, but still it can be stable because it is made up of bilayer. So the hydrophobic tails, they are protected by the hydrophobic interaction of the other monolayer tails. Okay, so that was explained, but it was still rejected because it could not explain the other properties of membrane. So it needed a little bit of upscaling. Then came the Davison and Daniel model. So this they stepped their game, and they stepped up their game, and they said that the membrane is actually a protein lipid sandwich. So that the sandwich looks something like this. So they said that there is a protein layer above and below, and between those two protein layers, lipid is sandwiched. So this is how the model should look like. But again, even though they explained the stability, now hydrophobic tails, that was again protected. Hydrophilic environment was again protected, but this was again rejected. This model was rejected because it had a major drawback, that during a lot of procedure we saw, during the experimental procedure, that lipases, those enzymes, they could easily digest the lipids. So lipases, these are the enzymes which can digest lipid. But if this model suggests that the lipid bilayer, or whatever lipids, they are, protected by the proteins, so this does not explain the digestion by lipases, because logically, if the lipids, they are being protected by the proteins from above and from bottom, so when we introduce lipase in the solution, they should not degrade the lipid, but they degrade the plasma membrane and disrupt the cell contents. This, because this was already proved experimentally, and that's why this model was rejected, because by logic this model says that lipases shouldn't, that they shouldn't degrade plasma membrane, but in reality lipases do degrade membrane, and that is why Davison-Danieli model was rejected, even if it explained the stability of model. Then came the Robertson model. So in Robertson models, was a bit advanced one. So in that, they stained the membrane with osmium tetroxide. So this is a stain. So when stained, so they were the first one to actually observe the plasma membrane. So when osmium tetroxide staining was done, they observed the membrane and they saw that it looks somewhat like railroad. So it has, the lipids are arranged in two rails, and the proteins, they are perpendicularly arranged to the lipids. Okay, so they explained that it looks like a railroad. So they showed that the membrane, so by that point of time, the membrane thickness was known, and they said that the membrane thickness is about around 6 to 8 nanometer, because it was evident. But when the protein extraction from the membrane was done, it showed that the membrane has different types of proteins, like globular proteins, and then beta sheet, big proteins like beta sheet proteins, beta barrel proteins, and it had a lot, membrane has a lot of proteins. Okay, and membrane thickness was 6 to 8 nanometer. So if you saw individually the size of lipid, it already occupies 4 to 5 nanometer, and then a protein, so then the space left to protein is only 2 to 3 nanometer. But the protein extraction protocols are already, it already evidently mentioned that it membrane has a lot of different kind of proteins, ranging from size of small globular proteins to small alpha helical proteins to larger beta barrel proteins. Okay, so membrane has diverse variety of proteins embedded in it. So how can a simple membrane with 6 to 8 nanometer dimensions can accommodate all those proteins? So this model did not make sense, even though it actually provided the evidence of this is how the membrane looks like, but it could not explain the structure well, and that is why it was rejected. Then came up the most accepted model, which is almost this is widely accepted, that was given by Singer and Nicholson. So it is also known as fluid mosaic model, because in this model it is it suggests that membrane is fluid. Okay, so it suggests that the membrane is kind of sea of lipids, a fluid of lipids, in which in between the proteins they look like icebergs. So it is a sea of lipids in which there are proteins here and there in between. So this was given by Singer and Nicholson model. So with this, I conclude lecture. In next lecture, we'll learn about this model, like why it said that it is fluid and how the fluidity of membrane was, proved. We will also learn in detail about the exact composition of models, how it is made up of phospholipids, the sphingolipids, and all. We will also learn about the functions of plasma membrane and the membrane proteins. So till then, see you. Bye bye.